Um, Good morning, good afternoon, good evening, and welcome. Julie Loisel and myself, Denis Didier Rousseau, are convener of this webinar, which is the second in a special series celebrating the 20th anniversary of Climate of the Past, which is an interactive journal of EGU. Today's topic is about high-resolution paleoclimate data, which provide detailed insights into past climate variability and extreme weather events on decadal to millennial scales. The recent past witnesses developing and refining methods to obtain high-resolution climate data from proxies like ice core, tree rings, and sediment layers, and selecting those for this webinar was really challenging. In theory, the webinar consists of two 30-minute talks and 30 minutes Q&A questions, with questions submitted only through the Q&A box at the bottom of the screen. Unfortunately, there was a last-minute change. Helle Astrid Kjaer talks was cancelled due to illness, which made us adjust the webinar timing, and we apologize for this inconvenience. Therefore, we move immediately to David Knauff's presentation. David is from the University of Bristol. He applies state-of-the-art isotopic and organic mass spectrometry to the study of lipids and molecular fossils, biomarkers, derived from organisms found in marine and terrestrial natural archives to understand the processes and mechanisms that drive changes in climate and biochemistry. His talk will speak about novel biomarker-based insight into the operation of the terrestrial methane cycle across the Cenozoic. So David, welcome, and the stage is yours.
Thank you very much. Thank you very much for the invitation, and welcome everybody again to this seminar to honor the 20-year anniversary of the EGU flagship journal Climate of the Past. So thanks to EGU, thanks also to Denis and Julie for inviting me. I will start sharing my screen. So I hope that works. Yes. Everybody can see.
Yeah.
Perfect. Yeah, so in the next 35-40 minutes, I got a bit of extra time because of Helle cancelling. I hope to share with you the research we've been doing here in Bristol, working on dynamics of the methane cycle, especially the wetland methane cycle across the last 68 million years. So going from, on the left here, the last bit of the age of the dinosaurs, the latest Cretaceous, across the whole Cenozoic towards the Holocene and modern day climate. And even we're going to look a bit into the future all the way at the end. So I hope there's something for everybody on lots of different time zones. But before I start, I just wanted to, before I start with the science, I just wanted to dive in a little bit into the reason why am I doing this or what? Because as some of you might know, lately I struggled, or earlier this year actually, I struggled with burnout. I was diagnosed with burnout, which has made me think pretty hard about what actually motivates and drives me in my job, and why are we doing, or why was I doing what I was doing. I'm now returning back to work, so I'm very happy to be able to present here what I'm doing. But ultimately, that was because I realized, like, I love finding out how the natural world operates, both in the present, the past, and the future, right? Whether it's from understanding microbial processes to reconstructing climate change across the past. I really love doing that, and that's actually why I ultimately chose a career in academia. So that's a big why. And another one is actually working with really amazing colleagues and PhD students and postdocs and undergrads across the world. I think as a job in academia, I also allows me to work with really talented people, and that's also one of the big motivations for me to do this. So I'm positive towards the future, and I just want to share some of the recent work we've been doing. A lot of this has been going on for the past few years, but some of the data is brand new. And let's talk about the methane cycle. If I can go to the next. Oh, here we go. So why study the wetland methane cycle, right? Why did I spend a lot of time on this? Well, the wetland methane cycle is a key part of Earth's system. Just methane in general is a very potent greenhouse gas. On the 100-year time scale, it has around 28 times greater global warming potential compared to CO2. We have added a lot of methane to the atmosphere as part of anthropogenic processes. And all this extra methane in the atmosphere has contributed to around 30%, so a third of all global warming that we have seen in the last 150 years, right? So roughly a third of that is because of methane. So methane is really important, right? And if we look at the natural sources of methane, you can see that basically all of it is terrestrial. You see freshwater lakes and wetlands are the two main natural sources, right? So I'm going to focus on wetlands because they are a main source of methane. And we also know that the methane concentration in the atmosphere, at least, is dependent on climate, right? The ice core data shows us, so here on the bottom, I show the ice core data with the methane concentrations of the last 800,000 years, so time running from the right to the left towards the past, and then on the right we go into modern day and actually a bit all the way up to present day values. And what you see is that over the last 800,000 years, we see quite big variations in methane from around 400 parts per billion during cold periods, called glacials, to around 700 parts per billion during warm periods, called interglacials. Right, and these variations, they might not look like that, they might look quite random, but actually these variations scale with climate. So when it's warm, we have higher methane, and when it's cold, we have less methane in the atmosphere. And in the last few decades mainly, and maybe 1.5 centuries, methane concentration have increased significantly. As you can see in the top panel here, they're still increasing year by year. So currently we are at levels of around 1934 parts per billion, right? So higher than anything we've seen in the past. So just to summarize that, methane is a potent greenhouse gas that plays a key role in the climate system, so it's important to understand. We know that variations in the terrestrial methane cycle are a key driver of glacial interglacial climate. We know there's a correlation there. But the problem is that we have almost no data beyond the reach of the Antarctic ice cores, right? So we have very little data spanning beyond 800,000 years. And although 800,000 years sound like a lot of time, actually for geologists, this is only a tiny fraction of all time there is, right? And lots of key things have happened further back in time, including past greenhouse periods, all of these. And we have very little data on how the wetland methane cycle operated during past greenhouse climates, right? So very little data, but we do have climate model simulations. A lot of this work is done by David Burling and Paul Vaudès, who look on very different time scales on like hundreds of millions of years, you should see in this. And these computer simulations now, where they model the wetland methane flux over time. And well, a few things to note there's lots of variation between different studies. But almost all studies indicate that either the methane cycle operated similar to today, the methane flux was the same, or it was order of magnitude higher, right? And there's big range on that. So modeling suggests it did operate different in the past, but again, we have very little data on that. And similarly, if we look at past greenhouse periods, like this is from the early Eocene, around 50 million years ago, these are again climate model simulations where they see how warm the planet gets if you just prescribe a certain amount of CO2, and you see that global surface temperatures and land temperatures are around 23 degrees Celsius, which is already much higher than we have today, fits with a greenhouse climate. But this is only using CO2. If they then add other greenhouse gases in these models, independent of CO2, so CO2 is the same, they just add other greenhouse gases, And with other greenhouse gases, most of that, around 60% of that, is actually methane. So a lot of it is methane. We actually see we get around 2 to 3.5 degrees extra warming, right? So again, in this past greenhouse climate state, it seems that methane could be a key driver, especially warming the poles. Methane could play a big role there, right? So from a modeling perspective, it seems like wetland methane cycle operated different in the past, but again, we have very little data on that. So the aim of my research over the past couple of years is to provide some insights on past dynamics of the wetland methane cycle, and that fits in with work that Rich Pencost here in Bristol already started around 2 decades ago, right? And in today's talk, I broke down in 3 different parts. I first want to talk about modern wetlands. Gives us a bit of a framework of what is happening in the modern day before going back in time. First, I will talk about the Holocene and the last deglaciation, so roughly the last 20,000 years, the period across which most of human history, at least all recorded human history, has taken place. And then at the end, I want to talk about longer time scale changes, looking at the Cenozoic and the latest Cretaceous over the last 68 million years. Right, so how are we going to do this? How do we provide insights into this key bit, the wetland methane cycle, right? Well, this is a very simplistic graph of a wetland where we have the atmosphere, and we have an oxic part of the wetland where we have plant growing, so peat vegetation, could be like sphagnum. And we have, below that, we have the water table, and below this water table, the peat is anoxic, so plants degrade very slowly and can then get decomposed. And these organic substrates can then be used to produce methane. There's like a key pathway to break down organic matter in peatlands. In the absence of oxygen and lots of other chemical species, basically methane production, methanogenesis, is a key breakdown product in peatlands. So at depth in peatlands, and this could be, this boundary between oxic and anoxic could actually be 10, 20 centimeters. Some peatlands, actually, the surface already gets quite anoxic. So it depends a bit where you are. But we have lots of methane being produced at depth. So we have a lot of this potent greenhouse gas being produced deep inside peatlands. And then there's different pathways this methane can take. This methane can be consumed anaerobically. So deep in the part can be broken down again. We think for most peatlands and wetlands, this is not a major sink. It seems to happen maybe in some wetlands, but in general, this is not a major sink. And actually, most of this methane produced at depth will diffuse upwards. So why do they diffuse upwards? Through plant roots straight into the atmosphere, right, will contribute to as a greenhouse gas. But a major control on how much methane actually makes it to the surface is, as soon as this methane hits the oxic layer, we can actually have bacteria that use oxygen to oxidize this methane. And this is called aerobic because it uses oxygen, methanotrophy because it's eating methane, right? And this is a major control. If you have more of this, that probably means there's more methane being produced. And then any methane that is not consumed makes it to the atmosphere, right? So the process I'm going to focus on in for the rest of my talk is this aerobic methanotrophy, right? So this major sink for methane in wetlands. That's the one I'm going to focus on. So it's important to note that we don't have a lot of data on any other of the processes, right? So we're just focusing on this one because for this, we actually have quite good tracers. So what are these tracers? So what is the method I'm using? So I'm using the delta 13C of hopanoids to trace methanotrophs. Now, that might be a lot of words that nobody understands. So I just want to break it down a little bit. What does it actually mean, right? So carbon has 2 stable isotopes, 12C, 13C. And we can measure this ratio and express it as a delta 13C. And if you measure different types of carbon, you can see that it has a very different carbon isotope composition. So here on the y-axis is delta 13C of different carbon species. So you see, for example, carbon in the ocean has a value around 0. You see atmospheric CO2 is a bit more depleted. Then you have plants and algae who are even more depleted because they're using this CO2 and they fractionate during this process. But the key bit is that methane because of how this is produced, is normally very depleted in carbon isotopes. Methane is quite unique in having a very depleted signature, often with values of -60 to -80 per mil, right? So that's delta 13C, and the key thing here is that methane is very depleted in carbon isotopes at very negative values. So what are hopanoids? So hopanoids are biomarkers. They look like this. So this is a carbon skeleton. It looks a bit like a sterol that we humans also produce. But bacteria produce hopanoids, looks slightly different. This is a hopene because it has a double bond. And lots of different bacteria make these lipids. And these lipids, this carbon skeleton itself is extremely stable. We can measure this in sediments as old as hundreds of millions of years. Is very stable, right? And the key thing is that this is produced by bacteria that eat methane, so methanotrophs, but also bacteria that don't do, actually, like heterotrophs, like that eating plant material, for example. And we can use the carbon isotopes of these carbon molecules to tell us something about the balance between the two. Because if we measure the carbon isotopes of these hopanoids, and we see it has a value roughly between -20 and -35 per mil, we see it's pretty similar to, for example, land plants and everything. We know that they're heterotrophs because they're eating this carbon. They're fractionating a little bit, but this means your pool of bacteria is dominated by heterotrophs. On the other hand, if your biomarkers have very depleted values like -50 to -80 per mil, that means they're consuming this very depleted methane, so your bacterial pool is dominated by methanotrophs. Now, this is the theory, but we also have direct data for that. For example, this is actually a really classic study from Roger Summons from the 1990s, where they grew bacteria on methane and then measured the carbon isotope composition of the hopanoids, in this case, diploptene. And you see here, you get these really depleted values of your hopanoids in these methanotrophic bacteria, right? So if you get very depleted values, that means that they're consuming methane. So we can sort of see this as a two-end member scenario. We can have a scenario where methanotrophs dominate the bacterial pool. These methanotrophs consume methane, which is very isotopically depleted. So that means that our hopanoids, our fossil markers, will be very depleted in carbon isotopes. And relative to biomass, normally values that are more depleted than -10. On the other hand, if we have heterotrophs dominate the bacterial pool, they consume organic matter, which has much higher values. So we have less depletion in the carbon isotopes, and relative to biomass, sort of like plus or minus 10 per mil. Using the carbon isotopes of hopanes, we can look at the balance between methanotrophs and heterotrophs, and we can say something about how active the wetland methane cycle is. And we have direct data from incubations. This is data from Julia van Winden, where she incubated sphagnum peat at different temperatures from 5 degrees to 25 degrees, then measured how much methane was being produced. And you can see at low temperatures, there's not a lot of methane produced. At high temperatures, you see a lot of methane being produced. And what they did in this incubation is they then measured the carbon isotope composition of both the vegetation, shown by this long-chain anocane, as well as the bacterial pool, in this case, diploptene. And what you can see is that when there's not a lot of methane, the bacterial pool has a carbon isotope composition that is very similar to the vegetation. So most bacteria are heterotrophs, eating the vegetation. But when you start to have a lot of methane at these higher temperature incubations, you can see the diploptene gets more negative, indicating that we start to have a contribution of methanotrophs. So when we have more depleted values, it's because there's more methanotrophs, because there's more methane. And what we normally do is we look at the difference between the anocane and the bacteria to correct for changes in the biomass. So this offset is what we're going to use, where an offset, a larger offset, more negative values means larger contribution of methanotrophs, more methane. So if you didn't understand anything I said so far, the main thing to understand for the rest of this talk is that more depleted carbon isotopes and hopanoids relative to biomass of this offset means there was more methane available. We had a more active wetland methane cycle. So now that we have a method to do this based on culture experiments, incubations, biological data, we have a method, we can start to apply this to the geological past. And people have done this in the past. But when we started on this project, we actually want to see, well, actually, how does it work in the modern day, right? We need a reference framework for the modern day. If we measure something that is 50 million years old, is that old? Is it not old? Well, we need to know how it looks in the modern day, right? So what we did is, and this is part of a large ERC, an advanced ERC project led by Rich Pankhurst and Gordon Ingalls and Jerome Bluer did a lot of the work here on this. There's also some data from a PhD thesis here that I added to this, looking at modern day peatlands. So all of these are like the top meter of peatlands, or the top 50 centimeters. I think it's the top meter. From across the world, although, as you can see, there's a bit of a bias to the Western world. We have no samples from Africa. So if anybody has modern day peatlands from Africa, drop me an email. Also, we need some from Australia, New Zealand, modern days. But what we did is in all these samples, we looked at the carbon isotopic composition of the hopanoids to create a modern day framework. What I'm going to show here, and I'm going to use this through the rest of my talk, is rain cloud plots. And if you want to know more about those, I suggest you look at this R package. It's a really powerful way of visualizing your data, and it goes beyond simple box whisker plots, has more information here. And what I'm going to show you in the modern day is the offsets between hopanoids and alkanes, where you have to remember that data plots in this band of plus to minus 10 per mil, that means heterotrophs are dominant, while if you have more depleted values, it means that methanotrophs starting to be abundant, right? And for this modern data as well, for the rest of the talk, I broke down the data into 4 different groups, into 4 different hopanoids. Hopanoids come in many different variations. I'm going to look at the short chain hopanoids, the hopenes, the ones with a double bond, and the C30 alpha beta and beta beta hopanoids. And the details are not really important. It's like all 4 of these groups sample a slightly different bacterial pool. So how does the data look like? So these are these rain cloud plots. So on the left, we have the short chain hopenes, so ones that have C30 or fewer carbons. Here in the middle, we have in green, we have the hopenes. So these have a double bond, C31 alpha beta hopenes. For those of you who are familiar with hopenes, but maybe not so much with peatlands, you might be wondering, why do we have C31 alpha beta hopenes in a modern day peat that is sometimes only a few years, decades, centuries old? Normally we find these compounds in very old, like 100 million year old rocks, but actually that's because of the acidic nature of peatlands, that this transformation of beta beta hopenes to alpha beta hopenes happens really, really quick. So peatlands are actually a bit unique that you have alpha beta hopenes in very recent sediments. So we have C31 alpha beta and C31 beta beta hopenes, and there's lots of details and nuances here that we can talk about, and I can probably spend a whole day on this figure, but I think there's some key things to see here. First of all, is that basically all the data plots within this blue bar of between plus and minus 10 per mil relative to biomass, indicating heterotrophs are dominating the bacterial pool. It doesn't mean that methanotrophs are not there. Just in the modern day, we see that heterotrophs really seem to be dominating at most locations. What we can also see is that the C31 alpha beta and the C31 beta beta hopane are a bit heavier, so have a bit higher values than the shorter chain hopanes and the hopenes, suggesting that in general, the source of these hopanes, and especially these hopenes, probably comes more from methanotrophs, while these C31 alpha beta and beta beta hopanes probably predominantly come from heterotrophs. But the key thing here, which is also the title of the slide, is that heterotrophy is king, right? Although we see a few hints of a few samples that are more depleted. And then you can summarize these rain cloud plots in these more simple box whisker plots here, but I will use for summaries later on. Right, so this is the modern day framework. We don't have every single modern day environment, but we're working on that, and we can expand. But in general, in modern day peatlands, we see heterotrophs dominating the bacterial pool. So now we have this framework for the modern day. We can actually look into the past. And the first one is looking at peatlands that span the significant part of the Holocene, and some of them even reaching back into the deglaciation. And this is a combination of work we're doing in Bristol, some unpublished work, working with colleagues from around the world. A lot of this has been published. Especially I want to highlight Yanhong Zheng, who's done a lot of work together with the Bristol crew on tropical and subtropical peatlands from Asia. But we're now also starting to have some data from other places. I also want to highlight the work done by Valentina Schaaf, who's done excellent work on tropical peatlands, especially from Africa, which is really important. So we currently have 8 locations across 4 continents where we have relatively old peatlands where we have data from. We have a total of 340 unique peat samples. And because you can often measure more than one type of hopanoid in each sample, we have more than 450 unique hopanoid carbon isotope values. How do these cores look like? Well, this is just the data range they span. So you can see some of them, like this core from Romania, actually reaches all the way back to 20,000 years ago. So it's quite an old one. While the one from James Bay Lowlands in Canada only spans the last 5,000–6,000 years. But most of them span at least most of the Holocene, right? And again, if anybody has some fancy old peat cores that are like 10–15,000 years old, drop me an email. I would love to discuss if we can analyze them and see if we can get a more global coverage, because as you can see, it's still a bit spotty. How does this data look like? So again, I'm showing you a rain cloud plot, so similar to the one I showed for the modern day, but now for the data from the last 20,000 years. And again, there's a few features to see. Again, we can see that the C31 alpha beta and beta hopanes are a bit have a bit higher values than the short chain hopanes and the hopenes. But I think the real feature to focus on is that we start to see these really depleted values, especially in the hopenes, right, with values that are as low as -25 per mil relative to the alkanes. So in absolute values, these are like -50, -55 per mil, which is indicating that we have a large contribution of methanotrophs during these, right? So we start to see some evidence of enhanced methanotrophy, some evidence of the wetland methane cycle operating different from what we see in most modern peatlands. Let's compare the data. So here on the left, we have the modern peatlands, and now here we have the deglaciation and the Holocene. And you can see a lot of the data is quite the same. The averages are actually pretty similar between those two. But the key bit is that we have a lot of these more depleted values. And some of this comes, for example, from China, where we see, for example, in this core, we see these really depleted values during a period between 4,000 to 6,000 years ago. And this is work done by our colleague Yanhong Zheng. And in this peatland, this is related to a drying event. And actually, every time we see these low values, we actually see that they're related to climatic perturbations. Right, so we see enhanced methanotrophy, more active wetland methane cycle during climatic perturbations. But if we then look on the global scale, right, so from the, from the 8 peat cores we currently have, there's no consistent timing or pattern across these, right? 4 of the 8 peatlands we looked at in the data that is available so far, we don't see any evidence of enhanced methanotrophy. We never see those really light values. But in 4 of these peat cores, actually, maybe it's not a coincidence, I don't know, but more of these Eurasian continental peat cores, we do see periods. But if we plot the timing of these, and these blocks indicate when in each of these peat cores we see evidence of very light methanotrophs or a more enhanced wetland methane cycle, we can actually see that the timing differs between all of them. In some of them, this happened quite some time in the past. So in Romania, we see this happening around 16,000 to 14,000 years ago. Well, for example, in China, we see this happening only between 4,000 and 6,000 years ago, right? So the key bit is that we see it happening every time there's perturbations, when we have transitions in wetlands, when we have drying events, but it doesn't seem to be a global timing, at least in the course we have looked at so far, right? And I just want to highlight that we are currently working on 2 cores from South America, one from Peru and one from Argentina. So we'll hopefully get a better tropical and maybe southern hemisphere picture as well coming soon. Right, so this is data for the Holocene and deglaciation. The next step is then to go further back in time and actually look at the last 68 million years. And for this, we're not looking at peat anymore. We're looking at lignite. So lignite is fossilized peat. Basically, it's peat that lost most of its water content, but it's still very thermally immature. So the biomarkers, most biomarkers are still preserved in there. The different, for example, from coal, which can lose certain biomarkers. So we focus on lignites. We currently have data from 30 different locations across 5 continents. Although again, large bias towards the Western world. We don't have any data again from Africa, and we only have data. We only have some data from Venezuela and nothing else from South America. So again, there are some gaps in the data. But we have analyzed more than 500 unique lignite samples, and some of this data has been published. So I show those here on the left in the references, both by Bristol, as well as other groups. And again, because we can measure often more than one hopanoid type in one sample, we now have a database of 1,300 unique carbon isotope values of hopanoids that spans the last 68 million years. These are all the locations and all the data. And again, lots of people have contributed, lots of people have sent us samples. There's been a lot of work going in developing age models. Really lots and lots of people, and I really want to thank all the collaborators that worked on this. Within Bristol, a lot of work on actually measuring the Hoppen isotopes have been done by Vittorio Laurentano, by Gordon Ingalls, and by Zhao Zhang, as well as other students and undergrads. You can see the data spans most of the Cenozoic. We have, for a lot of time periods, we actually have multiple samples from multiple places. So moving towards maybe sort of global signal throughout time. So let's look at the data, right? How does the data look like? So this is, again, these rain plots, but now for all these 1,300 samples from across the last 68 million years. And I will go in detail through this later, but I think there's a few things to notice here. First thing is, again, most of the data plots within this solid heterotrophy range. But we do start to see now some really light values, both in the Hoppenes and the short chain Hoppenes. And even in the C31 beta-beta Hoppenes, we start to see some quite depleted values. If you compare this to what we've seen in the modern and the deglaciation, if we look at lignite data, we can see that overall, all of the data is a bit lighter. We see a overall shift to slightly lower values. Also, the average is often slightly more depleted, especially in the C31 alpha beta Hoppane. It's a few per mils lighter. But most of it plots within this heterotrophy range. But we do have quite a few samples that actually indicate periods where methanotrophs really dominated the bacterial pool in those wetlands at time, indicating a really active wetland methane cycle. Now let's plot this data actually on age. So this is all the data now shown here in green at the bottom. And this is like this density plot to show you where we have data coverage. So you can see we still have a few gaps, but we have quite a lot of data, especially from this greenhouse world of the early Eocene. For reference, I show here on the top in gray the benthic oxygen isotopes, which show you a reference of climate. So we go from this warm climate state to a cold climate state over the last 50 million years, and also this reconstruction of CO2 across this time to give you some idea that we go from a greenhouse world to an icehouse world. But we see this cloud of data. Let's break it actually down in Hoppanoid type. Here we have the short chain Hoppanes across time. This is the data from the lignites. This is the data from the modern, and this band here plot like the range of the modern data with 2 standard deviations from the average. And I color-coded the data that lies outside of that in a slightly darker color. And what you can see is a lot of the data plots within the modern range, but we do have some periods where we see data that is much, much lighter. We see this especially during the early Paleogene, so around 55 million years ago, but also during the Cretaceous Paleogene boundary at around 66 million years. Now, next we can look at the Hoppenes. Again, a lot of the data plots within the modern range, but again, during the early Paleogene, we start to see these more depleted values. The C31 alpha beta hopanes. What is actually interesting there is that we again see lightest values during the early Paleogene, but it also looks like the overall data has shifted. If you look at the modern and all the data we have in the lignite, you see that basically all the lignite data is like 4 or 5 per mil lighter. So more contribution of methanotropes in general. Still a lot of it plots within this range of solid dominance of heterotrophs. And especially during the early Paleogene, we have some evidence of enhanced methanotrophy. And then the last one is the C31 beta-beta hopane. Again, most data plots within a modern range, but during the early Paleogene, we have these lighter values, right? So I think the take-home message is, so far is that a lot of the data plots within the modern range. So for a lot of the Cenozoic, we see values similar to what we see today, similar to what we see during the Holocene. But there are some exceptions where it looks like the wetland methane cycle operated different to what it does today. And let's actually zoom into some of those. So the first one is the Cretaceous Paleogene boundary. So this is the time when the non-avian dinosaurs went extinct due to a meteor impact. So you probably can't have a bigger perturbation of your climate system than smacking a massive meteor into planet Earth. This happened at 66 million years ago, and I just show here in red the benthic carbon isotope stack for reference. So we have this maximum in carbon isotopes marking the KPG boundary, and we have worked together with colleagues from 3 different places that gave us samples from the USA, Howell Creek, from Canada, Rhodes Creek West, and from Moody Creek in New Zealand. So at least data from 2 different hemispheres. I may just focus on data from the KPG boundary and slightly before and after. It does look like something interesting is happening. In all these places, both in New Zealand, in Canada, and maybe even in Hell Creek, we see these really depleted values exactly where the KPG boundary happened, right? So this is the time the meteor impacted Earth and all the non-avian dinosaurs went extinct. So it might be like, wow, okay, during the KPG boundary, also the wetland methane cycle responded, which is maybe no surprise. We see in the Holocene, every time we have a perturbation, when we have a drying event, something, the wetland methane cycle responds. As I said before, you can't get a bigger perturbation than smacking a meteor into planet Earth. So maybe that's no surprise. But if we look now at data that is slightly older and slightly younger and zoom out a little bit, the picture looks a bit different, right? And this probably also highlights why it is important to not just look at events and a few samples before and after, but actually look at the bigger picture. And what you see is that we also see very light values before and after the KPG boundary. And if you look like this, the KPG boundary actually doesn't stand out, right? So yes, we have perturbations during the KPG boundary, but it doesn't seem to be unique to just the KPG boundary. The next event I just wanted to spend some time on is the early Paleogene. So this is a period between 60 and 52 million years ago. This is often considered to be the warmest greenhouse period we have experienced in the last 66 million years ago. I show the benthic carbon isotopes in red again and the benthic oxygen isotopes in blue for reference. And the main perturbation stands out here around 56 million years ago. It's called the Paleocene thermal maximum. It's a very rapid, transient warming event, big perturbation, big extinction event for some organisms like benthic foraminifera. And again, when we look at hopane data, so we have quite a few data. A lot of this is published, but there's also new data here. When we look at data just before, during, and after the event, we see this massive perturbation during the PTM, right? We see very light values, as light as -40 per mil relative to the N-octane, so absolute values as low as -65-ish per mil, right? So there's really big perturbation here that doesn't look like anything we see before or directly after the PTM, right? And this is data from Ellesmere Island, where you again see that during the PTM, you get these really depleted values that start to look like unlike anything we see in the modern. But again, here it's important to look to put things into context, right? Because we look at it in the context of all the data that spans this period, you can actually see that there's similarly light data before and after. And even at the single location, and this is Ellesmere Island, so we have several lignite sequences there. You can see that also during the early Paleogene, this is the latest Paleocene, so before the PTM, we actually have quite depleted hopanoid values. sometimes more depleted than during the PTM. And also afterwards, we have quite depleted values, right? So yes, during the PTM, we see the wetland methane cycle operating differently, but it's not just unique to these known climatic events. Lastly, I just want to talk about the Neogene, the last 20 million years. And what is actually interesting is that when we look at all the data, we see this quite significant negative trend towards more depleted values over time. The closer we come to the modern, the more negative the values get. And the trend is around 1 per mil more negative per million years, right? So across this period, we see like a change of around 10-ish per mil, right? And I'm still not completely sure what is actually driving this, but one of the things that we know is happening across this period is this expansion of C4 plants, right? There's like this classic paper from Serling et al. From the early 90s, where you see this big change in the carbon isotopes of mammal tooth, going from organisms, horses, for example, and mammals dominated, that mainly are eating C3 plants, to mainly eating C4 plants, right? So you see this rise of C4 plants across the latest Miocene, starting or occurring roughly between 5 and 10 million years ago, when we see this shift, right? And we know from modern day experiments that the methane flux in C4 wetlands is much higher than it is in C3 wetlands, right? This is a logarithmic scale here. And you can see when you have almost no C4 vegetation, you have a much lower methane flux than when you have a lot of C4 plants, right? So potentially this shift here to more negative values, a larger contribution of methanotrophs, might be driven by the fact that we start to see a bigger contribution of C4 plants to wetlands, especially during the Miocene. So with that, I'm gonna list some conclusions. So I hope I've convinced you that as an organic geochemist, we can use the carbon isotope composition of biomarkers, in this case hopanoids, to reconstruct a key part of the methane cycle, focusing on the wetland methane cycle. I also hope that the data shows that the balance between methanotrophs and heterotrophs has been relatively stable over geological time, with a dominance of heterotrophs for most times. But we do see enhanced contribution of methanotrophs during key perturbations, like drying events during the Holocene. We see across the KPG boundary, the PTM, and the expansion of these C4 plants, although we also see other perturbations that are not linked to well-known climatic events. So what are the implications for that? Well, I think for modelers, like if you want to model past climate states, it's probably okay to assume modern wetland dynamics for a lot of the Cenozoic, right? Because our data shows that the carbon isotopes of the hopanoids are similar to today. But there are some exceptions, for example, like the PTM. But also what we see is that in the past, whether it's the Holocene or whether it's the PTM or the KPG boundary, when we have a perturbation of the climate system, we see the wetland methane cycle respond, right? And what we're currently doing with anthropogenic climate change is, of course, perturbing the climate system. So I think our data does suggest that this will have an impact on the wetland methane cycle, right? And I just want to show some evidence of what currently is happening, because here in the UK, we are currently suffering a drought, which might sound weird to people across the world, but yes, the UK can be dry as well. It doesn't always rain. And actually, especially in England, we have very, very low soil moisture content. So this is data from the 10th of July from this monitoring, government monitoring system, where they show that especially across England, we have very dry, or in some places even extremely dry drought happening. And we can see this. This is just a video I made in the park right outside my house. You can see in June, quite green, quite lush, right? But across the last few weeks, we can see that this has been drying a lot. And currently, this is how this park looks like, right? So very, very dry. And we see this across England, right? And this will have an impact on dynamics of peatlands as well. If we're drying out peatlands, this will have an impact. And we actually have modern day data. So this is data led by Dan Singh, where she looked into first modeling how soil moisture content will change. So this is the historical period. And then we see at the end of the century under different future climate change scenarios. You can see even in a relatively mild emission scenario, you can see that soil moisture is going to decrease. And we also know from modern-day experiments that will have a big impact on the methane cycle, right? If you go from a very wet location with a high soil moisture content to a very dry location, that has a big impact on methane, right? It can switch from a sink to a source. So all of this data does suggest that what we're currently doing with drying out our climate system, at least in parts of Europe, this will have a big impact on how the wetland methane cycle will operate. And with that, I would like to thank you for your attention.
Great. Thank you very much, David. I think it was very exciting. There is already one question in the Q&A box from Raj from Nepal. What could be the proportion of methane released from glacial meltwater, which may have old organic carbon in it? Can we estimate it through your biomarkers?
Yeah, that's a good question. I mean, old methane release is definitely important. We can't, with carbon isotope, like the simple carbon isotopes, the stable carbon isotopes, we can't look at old or young carbon, right? You could look at radioactive carbon, looking at 14C. That could be. I'm not sure how much of this methane would actually be fed into wetlands itself, but I mean, if you look at, I mean, and lots of people are doing this, like right, the groups of Tim Ecklinton, for example, is looking at measuring radiocarbon in biomarkers to see actually what is happening to this old carbon stored in permafrost and all of that. What is actually happening? That is that being consumed in rivers? Is it being does it reach the atmosphere? Is it being buried on the shelf in the Arctic, for example? So yeah, stable carbon isotope, what we are doing, this is probably not the method for it, but definitely looking at what the age of methane, where some of this methane coming are important research questions that you can answer with slightly different methods.
Julie, do you have any question? Yeah.
Yeah, I have a question. Thank you, David, for your talk. I always learn a lot from you. So you present a lot of data from the recent past and the Holocene from peatlands, and maybe it's fair to say that the much older samples you have are maybe beyond what we call peat, right? Would be more like wetlands. So my question would be, do you think you're missing a part of the opanoid signature because you're just looking at peat versus wetlands more broadly?
I mean, the whole definition, what is a peatland, what is a wetland? I mean, the same in the modern day database, right? I mean, we also have, for example, data from the Everglades in there, which might be more like a wetland than a peatland. So at least in the modern day database, we do have wetlands as well in there. For the peat cores of the Holocene, that's true. Those are all pretty solid peat cores. It's true, although a lot of them have transitions in them because they might, as you very well know, Julie, of course, like how they didn't start as a acidic peat bog, so there is some of that. But yeah, in the modern range, we do also have data from wetlands. But the thing is, in lakes, I mean, Rich and me always say lakes are weird. In lakes, you often see very depleted hopanoids because methane. The methane cycle in lakes can be very active, and in lakes you get really, really often very light hopanoids, right? So sometimes a bit of we don't want to reach the lake stage often, because then by definition we will get really depleted values, right? And of course, in some of our lignites, the argument sometimes is, well, actually, what is a wetland? What is a lake? An organic rich lake? And some of that sometimes can be tricky to interpret. So I fully agree with that.
Okay, but okay, cool. That was a good answer. Thanks.
So another question in the Q&A box from Vivian. So hi, thank you very much for your talk, David. Fascinating talk. She was wondering, how can isotopically light delta C13 excursions in sedimentary records be confidently attributed to ancient methane emissions rather than alternative carbon cycle perturbations? And what geochemical fingerprints distinguish microbial from thermogenic methane in the stratigraphic archive?
Yeah, no, it's very good question, Vivian, and thank you for that. So I think the first one is to look at whether the methane comes, whether it's methane emissions driving it or carbon cycle perturbation. So in all the figures I showed you, we look at the offset with the anocane. So for example, during the PTM, we know there's a big carbon cycle perturbation. And basically all alkanes get like, well, all carbon in the Earth, ocean, atmosphere actually got like 5-6 per mil, well, depends a bit which one you're measuring, but around 5 per mil lighter, right? But we correct for that by also always looking at the offset. So the alkanes across the PTM get like 5 per mil lighter. That's actually a key way to identify the PTM. And we look at the offset there. So we are looking at just the contribution between methanotrophs and methanotrophs. But I think an important one that Vivian is highlighting is actually the carbon isotope composition of methane can also change, right? As the Peat researchers know, methane can have wildly, it will be depleted, but it might be -50 or it might be -75, right? And if you change the carbon isotope composition of the methane, you will also change the carbon isotope composition of the hopanoids, right? I think in general, we showed light values indicates methanotrophs being dominant, and I think that's solid, but you can have variation within there just by changing the carbon isotope composition of the methane, right? So that's another lever you can turn on, right? But then you already are on the depleted side. But that probably matters why do you have values of -50 versus -60 per mil, right? But there are different levers you can turn or pull.
So I have a personal question.
Yeah.
You have been speaking about the wetlands, but what about paleosoil? Is there possibility to?
I have never looked at paleosoils, so I don't know. I mean, we do have lots of paleosoils.
Yeah.
I don't know. The good thing about peatlands and lignites is that they are full of organic matter. So it
Yeah.
makes my life as an organic geochemist relatively easy because it's easy to get the data out. Like, I mean,
Yeah.
sometimes I also work on carbonate-rich marine sediments, and that's a very different thing. Well, like for PGO. So I don't know if in paleosoil there would be enough organic matter to actually extract the hopanoid you would get. You could measure, but I mean, that would be an interesting thing to do. I don't know. Yeah, that would be an interesting, but I've never looked, and I'm not familiar with anybody who has done organic geochemistry on paleosoils, because from what I know, a lot of paleosoils are pretty thoroughly oxidized.
Yeah.
They don't have a lot of organic matter left.
Yeah, but okay.
But it would be an interesting project to see if there's anything we can see there, right? But on the other hand, the carbon isotope composition in paleosoils, it might also depend on respiration and lots of things. Like, I don't know. It might just be a way more complex system, and maybe methane is not one of the key variables there.
Yeah. Julie, have you another question? Because on the Q&A box there is none. So
I'm good, thank you.
I mean, it's almost 4 o'clock. Well, for me, it's almost 4 o'clock. I guess in Western Europe, it's almost 5 o'clock. So people want.
No, it's okay. But just going back.
Oh, there's a question in the Q&A.
Yes. So thanks for the great talk. Could you please talk a bit more about how has methane cycling changed across the intensification of Northern Hemisphere glaciation 3 to 2 million years ago, and is it relation to summer monsoon?
Yeah, I mean, this is a good question. The thing is that lignites need some time to develop. And I think the youngest lignite I have on my database, and I have to look at this, is like 3 million years old. Which is from Iceland, which is a very young one. And we don't have a lot of lignites that are Pliocene age, probably Pliocene. You probably can still look at this. There are some peatlands or preserved peatlands that are not turned into lignites yet, that are that old. So that would be an interesting one to look at too, because at the moment we basically have Holocene deglacial data, and we have, I would say, like the Miocene and everything all there. We don't have a lot of, well, we don't have any Pleistocene data, and we don't have a lot of Pliocene data either, just because there's not a lot of lignites available from that time period. But that would be interesting, because the summer monsoon change in those should have a big impact on where wetlands are, how they're operated, and everything. So I think that would be a really interesting one to focus on. And if people have good samples that span this time period, that would be really interesting to look at.
So just to follow up what you just mentioned, so you are missing Pleistocene peatland samples, but just for you to know that in a loose sequence that I have been studying with my colleagues in Germany, we have peat in loose deposits,
Right.
which are from the last climate cycle, and
Right.
it's so maybe it could be interesting for you.
That would be interesting. It would be interesting to bridge the gap, basically, right? I mean, because there's we can have some snapshots of at least Pleisto, whether it's lignite or peat, and that would be definitely interesting to look at. We just
it was very amazing when we found this peat deposits. It was corresponding to a thermokarst during glacial time. So it's something you could be interested with.
Would be interesting to look at. Yeah, exactly. At the moment, I've mainly focused on the older, on the lignites, but spanning the gap between the two, that would be quite interesting to do. Yeah.
Yeah,
so, Julie,
and I'll add to this, David, that Claire Treat had published a paper about peatlands from stage 5E. So a bunch of snapshots of old peat samples that were found all over, mostly North America, if I remember. But it could be worth looking back at those sites.
That would be really interesting. I think there is, I think I remembered, I think there's a peat core from Greece or something that spans the last 1 million years with like snapshots of it. So there is potential for all the peats. So much more work to do.
Okay, so Julie, do you not have any more questions?
No? Oh, good, thank you.
No, I think that we will close the webinar now. And so thank you again to David for this stimulating presentation, and thank you all for attending and for the interesting questions which has been asked. I would like to remind you that the webinar series is being recorded, and all webinars will be made available on the EGU dedicated page, thanks to Simon, which is behind the stage. The next webinar is scheduled for Wednesday, September 10 at 5 p.m. CEST. It will focus on greenhouse gases and climate sensitivity, and will feature two exciting presentations by James Ray and Jessica Tierney. So registration is now open, so please rush on the EGU page, and have a great summer. Thank you all.
Thank you.
Bye-bye. Thank you, David.
Bye-bye.